Fermentation is one of the oldest and most inexpensive means of producing and storing food. It is suspected that food fermentation for the preservation of cereals and legumes originated in China. The practice has been extended to many other foods including vegetables, fruits, edible parts of plants, fish, milk, and meat. According to Vijaya Lakshmi, food fermentation is a way of supplying the necessary quantities of foodstuff instead of preserving and sustaining existing foodstuff or enhancing their consistency. Fermented rice, also known as tapai, is a fermented food with a starter culture formed from inoculating a carbohydrate. Tapai is known as traditional fermented preparation of glutinous rice and known as dish served mostly in East and Southeast Asia. Tapai is made by fermenting glutinous rice. Ragi used in the experiment act as fermentation starter. Ragi tapai is a dry starter culture prepared from a mixture of rice flour, spices, and water or sugarcane juice or extract. The fermentation requires microorganisms in order to convert the starch into glucose. The resulting paste has a sour and sweet taste and is mostly directly consumed as food or used in traditional recipes from East and Southeast Asia. The objective of this experiment. First, understand the fermentation principle of the solid substrate used to transform the local starch sources. Second, to learn the method to prepare tapai and the technique during the preparation. Third, to identify the effect of fermentation process on physical, chemical properties of tapai. Fourth, to get to know the Malaysia fermented foods and their background. What is So, do you guys know how the process of tapai making? Okay, let's watch together. In conclusion, fermentation is the conversion of carbohydrates to acid or alcohol. Thus, this experiment had to observe the aroma and flavor of fermented products. Tapai made from this experiment give the result which are good flavor, color, aroma, and appearance. Furthermore, alcohol produced also play a vital role to extract flavor components from substrates. The longer tapai are fermented, the higher are their acidity content. It is because alcohol content will increase day by day during fermentation process. A fermenter is operated by inoculating a complex sugar medium with a microorganism. This microorganism is generally allowed to reproduce under aerobic conditions before the fermenter is switched to anaerobic conditions to produce secondary metabolites such as ethanol. In this fermentation of tapai, the substrate becomes soft and some acid are formed as shown by the soft alcoholic smell produced. The fermentation requires microorganisms in order to convert the starch into glucose. These experiments also help to identify what microorganisms occur during fermentation process. There are a few types of microorganisms in yeast such as Saccharomyces cerevisiae and Saccharomyces fibulata, and those are used to convert glutinous rice into tapai.